Hi students, welcome again to part 2 of composition of blood. Now we'll continue the topic, the composition of blood. In the last topic we have seen the blood plasma. Blood plasma क्या है? What are the main components of blood plasma? Like water, mineral ions, those fluctuating and constant components. Now if you have missed that video, the link is given over here. आप वो वीडियो देख सकते हैं. Blood cells. इस वीडियो में हम देखेंगे blood cells. Now there are three types of cells. These are also called as the formed elements or free cells. As we all know that we have seen in the last video, it is 45% of your blood. Now there are three types of bloods. What are those three types of blood cells? The first one is called as the erythrocytes or red blood cells. इसको हम लोग RBC कहेंगे. The second is the white blood cells or leukocyte, इसको हम लोग WBC कहेंगे. And the third type is platelets or thrombocytes, इसको हम लोग platelets ही कहेंगे. Red blood cells or RBCs or erythrocytes, they help in oxygen and carbon dioxide exchange, means gases exchange, respiration basically. The white blood cells or leukocytes helps in fighting against the infection. They form the cellular immune system of your body. And the platelets or the thrombocytes helps in blood clotting along with the fibrinogen, जो हमने लास्ट वीडियो में देखा था, the plasma protein fibrinogen and prothrombin. तो platelets help in blood clotting. So हम यहाँ क्या देखने वाले हैं? We are going to see how these cells are originated, what is the speciality of these cells, what is the structure of this cell, how they can be differentiated from each other, and what is their function. So let's start with the RBC or the erythrocytes. RBC or erythrocytes. See, these are the most prevalent of all these cells which are found in the blood. Now if you take a blood sample and if you take a drop of blood I am saying, you can find around 5 millions of these cells in 1 cubic millimeter of your blood drop. एक cubic millimeter आपके blood drop, 1 millimeter का एक cube, 1 millimeter by 1 millimeter by 1 millimeter by 1 millimeter, आपका square होगा, 1 millimeter का square होगा. You'll find there are 5 millions of these cells. These cells or are non-nucleated or enucleated. Nucleus नहीं होता है इसमें. Nucleus नहीं होगा तो DNA नहीं होगा. DNA नहीं होगा, साथ में कोई भी प्रोटीन सिंथेसाइजिंग मशीनरी नहीं होगी. तो इसका मतलब है कि ये सेल प्रोटीन सिंथेसिस करता नहीं है. इट इज नॉट इन्वॉल्व्ड इन द प्रोसेस ऑफ प्रोटीन सिंथेसिस लाइक एनी अदर सेल. जैसे नॉर्मल सेल्स में होता है, डीएनए होता है, फिर डीएनए का आरएनए बनता है, प्रोटीन सिंथेसिस होती है. यहाँ पे ऐसा कुछ नहीं होता है. तो इसमें होता क्या है? लेट्स सी द स्ट्रक्चर ऑफ दिस सेल. इफ यू लुक at the structure of an RBC, as I said, it is a non-nucleated spherical disc. एक non-nucleated spherical disc है. Now this is only in case of us humans, in mammals. In mammals, only in case of humans. Lower vertebrates में इसका structure oval होता है with a small nucleus also. In case of mammals, only one mammal shows presence of an oval nucleated RBC, that is the camel. Now this picture what you see over here is an RBC. This picture is an RBC of a camel with presence of a very distinct nucleus, whereas in case of humans the nucleus are totally absent. So there must be some condition. Now because of the living condition of the camel in the desert, the RBCs which are oval in shape, they also help in retaining the water. So ये swell up हो जाते हैं जब भी camel gets water, the blood में water level जब बढ़ जाता है तो RBC swell up and they retain the water, which is very much important to survive in the desert condition. And this is one very important adaptation the camel shows to adapt or to survive in the desert. Structure देखते हैं हम RBC का. A normal human RBC it is around 7.2 micron, 7.5 micron in diameter. So this the diameter of the RBC from this point to this point is around 7.5. The thickness, it is around 2.4 at the edges, and in the center is around 1. So it is actually less thicker in the center and more thicker at the periphery. So the structure of the RBC is somewhat like a disc, which is flattened in the center and thicker on the periphery. You can compare it like a मेंदू वड़ा, है ना, बस उसमें होल नहीं है, but it is very much thinner in the center and thicker at the periphery. The RBCs are acidophilic. Acidophilic means they take up acidic dyes or acidic stain. One of the acidic dye or stain which we use is eosine. So जब भी हमको RBCs को देखना है, we use eosine stain to stain them and to see them. Other than that, endoplasmic reticulum नहीं है, Golgi complex नहीं है, mitochondria absent है. Means other than the presence of hemoglobin, इसमें सारा का सारा cellular organelles absent है. DNA नहीं, nucleus नहीं, endoplasmic reticulum नहीं, Golgi bodies नहीं, mitochondria नहीं. इसका मतलब there is no protein secretion, there is no protein synthesis going on. What is present? Around 64% water है, 28% hemoglobin है. यही hemoglobin, it is actually a respiratory pigment. And because of the hemoglobin, the RBC gets its red color. Around that, we have 7% of lipids in it, 3% of sugar, salt, enzyme, and other protein which is present or which the RBCs get during the process of formation of the RBC. What is the process of formation of the RBC? पहले फंक्शन देख लेते हैं इसका. फंक्शन क्या है इसका? एक ही फंक्शन है, HB यानी हीमोग्लोबिन का फंक्शन है ऑक्सीजन ट्रांसपोर्ट करना. So it gets oxygen from the lungs, the blood goes to the heart sends that oxygenated blood to each and every organ. So the blood goes to each and every organ. Over there the diffusion of gases takes place. So oxygen is given from the hemoglobin to the tissue, and that's how the tissue gets the oxygen. It also helps in excretion of carbon dioxide. Yes, excretion means putting some waste material out of the body. It also helps in excretion of carbon dioxide, a small quantity of carbon dioxide from the lungs. इसका लाइफ हिस्ट्री देखते हैं. लाइफ हिस्ट्री क्या है आरबीसी की? ना न्यूक्लियस है, ना प्रोटीन सिंथेसिस है, फिर भी हमारा आरबीसी अराउंड 120 डेज जिंदा रह लेता है. एवरेज लाइफ ऑफ एन आरबीसी इज 120 डेज, 120 to 125 डेज तक रह लेता है अंडर नॉर्मल कंडीशंस. एंड इवन इफ इट इज डिस्ट्रॉयड, at every minute around 140 million RBCs are produced in your body. So don't worry, 120 दिन बाद अगर RBC मर रहा है तो every minute you are getting 140 million new RBCs which are formed in your body at the same time. In case of embryonic condition में जब RBC प्रोड्यूस होना स्टार्ट होता है या RBC प्रोडक्शन स्टार्ट होता है, it happens in two organs. One is known as the liver, another is the spleen. Now what happens till the embryo gets birth? जब तक embryo पूरी तरह फॉर्म नहीं होता, liver and the spleen produces RBC. But in the adult condition, this production of RBC is limited to only one site in your body, that is called as the bone marrow. Bone marrow is the tissue, the red blood cell tissue present in the cavities of your long bones of your body. It could be the long bones of your hands and the legs and the ribs, and this small connective tissue in your cavities of your long bone is called as the bone marrow. Production of RBC is called as hemopoiesis or erythropoiesis, and this RBC or the hemopoiesis and erythropoiesis occurs from a cell which is called as erythroblast. So the germinal cell, or you can say the stem cell, erythroblastic stem cell which हम लोग कहते हैं, the stem cell which is present in the bone marrow is called as erythroblast. This erythroblast differentiate into RBC and other blood cells. And how it differentiate? Differentiate under a factor, influence of a factor known as erythropoiesis stimulating factor. Erythropoiesis stimulating factor, which is called as erythropoietin. ये जो erythropoietic stimulating factor है, इसका नाम है erythropoietin. Erythropoietin acts on the red bone marrow or the erythroblast to differentiate into RBC or to initiate the erythropoiesis. Now we'll see when this happens and when the erythropoietin is secreted, why it is secreted, and how erythropoiesis starts. Now what you see over here is the liver. This is the liver, and this over here, small organ what you see is called as the spleen. No, in the adult condition, the spleen is not responsible for production of RBCs, but the spleen functions in storing the RBCs. ये जो मैंने कहा कि RBC lives for 120 days. What happens after 120 days? The RBC doesn't die that quickly, but it goes to the spleen and it is stored in the spleen for 120 days. Now, this spleen, when your body feels that there's a requirement of RBC, the spleen has to give those RBCs back, which are viable, which can be used. And for that, the spleen squeezes. And when the spleen squeezes, RBCs are pushed back in the vascular system or the blood circulatory system. That's why when you have blood loss or when you're anemic, there's a pain in the stomach because of the squeezing of the spleen. Okay. And this substance, the erythropoiesis stimulating factor or the erythropoietin, is also responsible for erythropoiesis. Now understand the system or understand the process over here. There is a bone marrow, a red bone marrow present in the long cavity of your bone, which has an hematopoietic stem cell. The hematopoietic stem cells differentiate into erythrocytes. The erythrocytes as well as the other cells like the platelets or the thrombocytes and all the WBCs or the leukocytes. These hematopoietic stem cells can also be differentiated into lymphoid progenitor cells. So they can also form the T cell and the B cell or the lymphocytes. We'll come to what are lymphocytes. And this hematopoietic stem cells, they show presence of the capacity to differentiate into more than 2 types of cells. This is called as pluripotency. Pluri means plural, pluripotency. They can differentiate into different types of cells. They are not totipotent, they are pluripotent. Kidney or our renal system also has a role to play in the hematopoiesis. You know, when there's hypoxia or when the oxygen concentration in your blood lowers down, जब oxygen concentration कम हो जाता है. Now, when this happens, suppose if you are living in a condition where the oxygen density is very low, say for example if you go to a higher altitude, जब हम higher altitude में चले जाते हैं and the oxygen concentration is low, या density of air low है, and then we feel that we need more oxygen or we cannot breathe that easily, kidney feels that pressure and kidney needs to induce this process known as the erythropoiesis. For that, the kidney actually secretes. Now kidney over here becomes a secretory organ and the kidney cells secrete erythropoietin, and that erythropoietin is secreted in the blood. That blood goes to the bone marrow, and under the influence of this erythropoietin or the erythropoiesis stimulating factor, the bone marrow starts producing more RBCs. So RBCs are produced more. This also happens if you're suffering from iron deficiency. So ज़्यादा आरबीसीज बनना चालू हो जाते जब आप कहीं पे हायर एल्टीट्यूड में चले जाते, हिल स्टेशन में चले जाते, टू कंपनसेट दैट लो ऑक्सीजन कॉन्सेंट्रेशन। तो जितना ऑक्सीजन कम होगा उतने ज़्यादा आरबीसी होंगे, तो कंपनसेट हो जाएगा। एंड दैट्स हाउ योर cycle of erythropoiesis also happens. So when the number of RBCs increases, the kidney stops secreting erythropoietin, and then your blood volume remains constant. That's why when you go to a hill station, initially you feel a little bit difficult to breathe, but after some days, after 2 to 3 days, your body adapts itself to that level of oxygen by having more number of RBCs. And if you have your test done over there before going to hill station and after reaching to hill station after around a week, you'll notice that the number of RBC या फिर आपका RBC count बढ़ गया है या erythrocyte count बढ़ गया है. This is like your feedback mechanism. आपके बॉडी आपकी खुद की बॉडी को फीडबैक दे रही है. See, ऑक्सीजन लेवल कम हो गया, you need more RBC लेवल बढ़ गया, चलो अभी RBC बनाना बंद कर दो. क्यों बनाना बंद कर दो? सपोज बनाना नहीं बंद किया तो क्या होगा कि आपके बोन मैरो में कांस्टेंट जनरेशन चलते रहेगा आरबीसी का, नंबर ऑफ आरबीसी बढ़ जाएंगे. तो दिस इज अबाउट द प्रोडक्शन और द सिंथेसिस या फिर जनरेशन ऑफ आरबीसीज हम कहेंगे. व्हाट हैपेंस व्हेन द डिस्ट्रक्शन ऑफ आरबीसीज हैपन? तो आफ्टर 120 डेज जब आरबीसी की जरूरत नहीं तो क्या करना है उसे? से डिस्ट्रक्शन हैपेंस इन द लिवर, तो आरबीसीज गोस टू लिवर, एंड ओवर हियर नॉन सिलेक्टिव प्रोसेस है ये, कोई ऐसा कोई नहीं कि सिलेक्टिव है, रैंडमली हो जाता है, नॉन सिलेक्टिव प्रोसेस है. दी आरबीसीज आर डीजनरेटेड और डिसजनरेटेड, डिसइंटीग्रेटेड, और जो भी एचबी निकलता है, हीमोग्लोबिन उसमें से निकलता है, दैट इज मेड अवेलेबल फॉर द मेटाबॉलिज्म, इन द फर्दर मेटाबॉलिज्म, दैट इज रीयूज्ड अगेन फॉर न्यू आरबीसीज. और दैट cytochrome, there is a component known as cytochrome, okay, from the hemoglobin is used or it is destroyed and it is excreted out of your body in the form of bile or in the urine. So bile is responsible to give coloration to the urine. That is, if there is more amount of bile, your urine becomes more yellow. If the bile is less, urine is less, or you can say faint yellow in color. And that's why even the volume of blood or volume of water in your plasma also affects the color of your urine. So ज़्यादा अगर destruction of RBCs हो रहे हैं तो bile secretion ज़्यादा होगा, और अगर destruction of RBCs कम है तो bile secretion कम होगा. यही हम देखते हैं jaundice में. Jaundice is the infection of liver, and over here if more of the destruction of RBC is happening and more bile is secreting out, you will find the blood has more bile concentration, and because of which your organs become yellowish in color. आपकी skin थोड़ी सी yellowish हो जाती है. That's the reason. So that is about RBCs. ये कुछ सीजन और आगे जाएगा. This is a lengthy topic. तो हम अगले सीजन में मिलते हैं, पार्ट 3 में मिलते हैं. ये भी हमारा कुछ सेकंड सीजन में खत्म नहीं हो रहा है. सो पार्ट 3 की लिंक इस वीडियो की यहाँ पे दी गई है. ओके, तो देखिए वीडियो और आगे बढ़ेंगे. ठीक है, थैंक यू.